Great. Good morning, everyone, and welcome. On behalf of all of us at the Boston Globe, I am thrilled to welcome you both in person and virtually to our second annual Future of Medicine Summit. My name is Lynn Henry. I have the honor of serving as the CEO of Boston Globe Media. Today, we gather at a moment of extraordinary change. Medicine is being transformed before our eyes by science, by technology, and the visionary people who push the boundaries of what's possible. This is also a transformative time with rapid major policy changes and major funding cutbacks impacting the future of medicine in real time, as the newsrooms of the Boston Globe and Stat are reporting on each and every day. So, given all of that, I cannot imagine a better time for us to convene some of the great thought leaders and practitioners here so we can think, process, and learn together what all of this means. Here in Boston, we're fortunate to be at the very center of the innovation in medicine. Our city is home to world-class hospitals, research labs, universities, and companies that are not just participating in this evolution, but leading the world into a new era of health and discovery. At the Globe, our mission has always been to illuminate, to connect, and to hold up the stories that matter most to our community. And that's why events like this are so important. They bring together our community and leaders to share knowledge, ask hard questions, and imagine together what a future might look like. And what a great program we have lined up for us today. We're going to hear from extraordinary scientists, clinicians, and speakers, from a breakthrough prize winner to pioneers reimagining cancer treatment, aging, and metabolic health. We're going to explore how innovations like Ozempic are reshaping medicine and markets, how young change makers are redefining the path forward, and how visionaries have set the foundation of breakthroughs yet to come. I want to thank today's moderators, who are our brilliant Globe journalists who bring curiosity, rigor, and heart to every conversation. They'll also help guide us through today's themes and ensure that the ideas here don't just stay in this room, but ripple out to the wider community. A huge thank you to our event sponsors, Collegium Pharmaceuticals and Harvard Pilgrim Healthcare, for making this very important event possible. And most importantly, thank you for being here today, for your support and engagement, and for bringing your curiosity here to today's conversations. Together, let's dive into the future of medicine. And with that, it is my honor to introduce our first guest, Dr. David Liu, a Breakthrough Prize winner, who will be joined by Globe reporter Anna Kuchman. Thank you, guys.
Hello, everyone. And hi, Dr. Liu. Thank you so much for joining us today.
My pleasure. Thanks for having me.
Dr. Liu, I want to start with, I'm sorry. Earlier this year, one of the gene editing techniques that you developed helped save the life of a 5-month-old baby named KJ in Philadelphia. And I was wondering if you could tell us about that case.
Sure. So first, and maybe most important, this is work led by a large team of researchers from many organizations, but really organized and led by Karen Musunuru and Rebecca Ahrens-Nicholas, both at Penn, Children's Hospital of Philadelphia. And the case involved baby KJ Muldoon. So this was a baby born with 2 mutated copies of a gene called CPS1. One inherited from mom, one inherited from dad. And if you have just one bad copy, you're fine. If you have 2 bad copies, you have a serious disease. About half of infants with this disease will die in infancy, and you suffer progressive brain damage. So Kieran and Becca quickly realized, based on a genetic diagnosis, what the problem with this baby was, as their blood ammonia levels rose to dangerous levels. And so what was done was the development of the first bespoke, customized gene editing treatment. This was a base editor that was literally designed specifically to correct one of the 2 mutations, happened to be the one that baby KJ inherited from his dad, changing the mutated letter, the G that causes the disease, back into an A, changing the A that causes the disease back into a G, which rescues the normal function of the gene. And the baby was dosed with this drug less than 7 months after diagnosis. The process of developing a new customized base editing medicine is something that I normally think of as taking maybe 7 years, not 7 months. So it was really a testament to the robustness of the science, the incredible leadership and organization of Kieran and Becca and all of the participating groups, as well as I think, good judgment of regulators to say this is a case where the risk benefit ratio clearly favors treatment because we know what will happen if the baby isn't treated. And so the baby was dosed less than 7 months after diagnosis and happily appears to be doing fine now, is reaching developmental milestones and eating a normal diet that uh, children with this disease normally can't reach and can't enjoy.
That's great. And the specific technique that you developed that was used was called base editing. Can you talk a little bit about how you developed that and how it works?
Sure. So to understand base editing, it first helps to begin with the breakthrough discovery of CRISPR. So CRISPR, as many people may be aware, is a bacterial immune system that evolved as a way of allowing bacteria to cut and disrupt the genes of viruses that infect those bacteria. So CRISPR uses a small piece of RNA to target a piece of DNA. And when it finds that DNA, it cuts it. And when you cut the double helix, you generally disrupt the function of that piece of DNA. So that's how CRISPR works in nature. However, for most genetic diseases, the most direct way to benefit the patient requires correcting the mutation, just as in baby KJ's case, rather than disrupting an already broken gene. And so that inspired us, beginning in 2013, but culminating in 2016, the development of the first base editor. So base editors don't exist in nature. They use that beautiful RNA programmed targeting mechanism to find a DNA sequence of your choosing, but instead of cutting that sequence to disrupt the gene, they instead perform chemistry on an individual base to convert, for example, a C into a T, or to convert an A into a G. And in some cases, nature doesn't even provide the enzymes needed to perform that chemistry on DNA. In those cases, we had to evolve in our laboratory the enzymes to do so. So base editors are kind of like DNA modifying pencils that change a single letter of your choosing into a different letter, and that can be very useful because most genetic diseases turn out to be caused most commonly by single letter swaps, single letter errors in DNA. So base editors can make efficiently and cleanly 4 different kinds of single letter swaps. And one of those was the single letter swap that we used to rescue baby KJ's CPS1 deficiency.
Great. And then you also developed another technique called prime editing. Can you talk about that as well?
Sure. So we reported base editors first in 2016, and then the second flavor in 2017. That's actually the flavor that was used in baby KJ and in many other clinical trials. There's actually more than 20 base editing clinical trials that have begun. And then in 2019, we reported the development of a different kind of gene editing called prime editing. Like base editing, prime editing uses the beautiful RNA programmed DNA homing mechanism of CRISPR, but also like base editing, instead of cutting a chromosome into 2 pieces to disrupt a gene, prime editing performs true search and replace editing, meaning it finds a target DNA sequence, writes a new segment of DNA that you wish to replace the original sequence with, and then guides the cell through DNA repair processes that result in that new flap of DNA replacing the original DNA sequence. And because it's search and replace, just as you do when you use search and replace in a word processor, you can make virtually any kind of local change in DNA using a prime editor. So you can change any letter to any other letter. There's 12 different ways you can change one of the 4 DNA letters to a different letter. You can make insertions of missing letters, like the missing CTT that is the cause of cystic fibrosis in most cystic fibrosis patients. You can remove extra letters, like the extra TATC, that is the cause of Tay-Sachs disease, the most common cause of Tay-Sachs disease. And very recently, prime editing was used in this search and replace capability to benefit patients with a debilitating and life-shortening immunodeficiency called chronic granulomatous disease, or CGD. So one of the most common causes of CGD is two missing letters, GT in a gene called NCF1. And so the scientists at Prime Medicine used prime editing to insert the missing GT at just the right place in the NCF1 gene to restore the normal sequence and therefore the normal activity of that missing enzyme. And so one of the first two patients treated was a man named Tracy Atterbury. And some of you may have seen the story in the New York Times. Am I allowed to say New York Times here?
Yes.
Okay. In the New York Times yesterday that described how this treatment seems to benefit Tracy, yet also raises this, I think, incredibly important issue that scientists, doctors, regulators, and really all of society has to face, which is these gene editing technologies, including base editing and prime editing, now allow us, in principle, to correct the vast majority of DNA misspellings that cause genetic disease. There are 10,000 genetic diseases that collectively affect about 400 million people. So, to put that into context, about 50 million patients live with cancer. They receive, on average, per patient, a global investment of $10,000 per cancer patient. There are about 39 million people that live with HIV/AIDS in the world. They receive a global investment in HIV/AIDS research of about $500 per person. There are 400 million people in this world that live with rare genetic disease. They receive a total global investment per patient of about $3. So it is, I think, one of the largest yet most underserved communities of patients in the life sciences and in medicine, in part because until these programmable gene editing medicines, there wasn't a way that one could imagine using a common technology to treat many of these different diseases. But now there is such a way, and we have to figure out as quickly as possible how to connect the science with the patients, because individually, these diseases, the vast majority of them, are too rare to make a profit. So a company that has a fiduciary responsibility to stay in business so they can keep developing medicines won't be able to take on the vast majority of these products. And so I think the answer ultimately is a combination of regulatory streamlining and flexibility, keeping in mind the patient benefit, the risk benefit ratio, having, as HHS has announced recently, a plausible mechanism to benefit the patient with some reasonable demonstration of safety. Uh, and perhaps in those cases where the patients stand to really benefit, it's worth streamlining this process so that the patients can get access to the medicines, even short of a traditional, 10-year development cycle that costs $100+ million, which will never end up being practical economically for a disease that might affect 1 or 10 or 100 patients, or even 1,000 patients.
Mhm. And can you talk a little bit about how widely available gene editing is now and kind of what the future holds? What are some future advances that we should be watching for
Right.
from your field?
So, in terms of the base editing and prime editing clinical trials, the ones that I keep most close watch over, there are at least 23 such trials that have begun. We publish all of our science in the public, so these trials have begun in a number of countries. In fact, there are more base editing and prime editing clinical trials taking place in countries outside of the U.S. than in the U.S., even though these technologies were developed in our lab. Um, and I think that just speaks to first the tremendous need. It's a worldwide need. It's not a U.S. need. Uh, but I think your question really highlights the key issue at this point. Uh, the science, in some cases, such as genetic liver disease, where there's a very well-defined association between a DNA misspelling and the disease, as in baby KJ's case, The science in those cases is not the bottleneck. We know how to even deliver these gene editing agents into the liver in ways that will very likely help or even save the lives of patients like baby KJ. Uh, Instead, it's really the system that needs to keep up with the science. We need regulatory streamlining so that when you change the little RNA program that represents less than 3% of the atoms of a base editor to send baby KJ's treatment to a different site in the genome to treat the next baby with a fatal genetic liver disease, that you don't have to start over, that you don't have to start completely from scratch and invest the millions of dollars and the new animal models and the new uh, biodistribution toxicity in a monkey study, et cetera, so that we can dramatically streamline these treatments, reduce the cost of development, which ultimately, of course, will reduce the cost to the healthcare system as well. It's already estimated that every genetic disease patient costs the US healthcare system between $2 and $10 million over their lifetime. So there is plenty of benefit, not just to the patients and their families, which should still be foremost in our mind, but even just to the US healthcare system for one and done treatments that can actually provide a lifetime uh, therapy for a debilitating disease. So the external factors all point towards the feasibility and I think the ethical mandate to connect this science with patients. We just have to evolve the non-science parts to keep up with the science so that the regulation, the manufacturing, the distribution, the reimbursement system, the collaboration between academic labs, nonprofits, and companies can all come together to do what we strongly believe is possible already, which is to save many more lives, like Alyssa Tapley's, like baby KJ's, like Tracy Atterbury, the patient treated with the prime editing medicine, and many others, because that is really this unique moment we're in now, where maybe for the first time DNA is not destiny, and if you happen to have a misspelling in your DNA that would normally um, create a life for you that is full of hardship or shorten your life, uh, that now there are things that you can do in some cases uh, to depart from that fate. I think it's up to many members of society to figure out how to best connect these capabilities with the patients who urgently need these treatments.
Mhm. And in terms of kind of the future development of the technique, until now, it's mainly been used for rare diseases, single and maybe double gene mutations. Is there the possibility for it to be used more widely for a much broader set of genetic diseases?
Yeah, absolutely, and you can see how the progression will likely take place. In fact, one of these 23 base editing and prime editing clinical trials underway is a trial initially started as a collaboration between Verve Therapeutics and Beam Therapeutics. Lilly then bought Verve Therapeutics. But the trial is to use an adenine base editor, the same flavor of base editor that was used in baby KJ, to install a single letter change in a normal gene called PCSK9 that everybody here has. We all have normal, probably normal, PCSK9 genes. But if you make a single letter change at just the right position in PCSK9, you will lower your LDL, bad cholesterol level, by up to about 2/3 permanently. And that level of LDL cholesterol lowering integrated over the lifetime of a patient is thought to have tremendous benefits in reducing the risk of coronary artery disease, which is the number one killer of humans in pretty much all countries right now. Um, so that trial is underway and has already been shown to be efficacious and reasonably safe in humans. Uh, and that's an interesting case. The first cohort of patients that were given this treatment are those that have absurdly high LDL cholesterol levels and as a result are at grave risk of heart attack or stroke. These are people who typically have other kinds of genetic disease that cause their LDL cholesterol to be, 300 or something very, very high like that. But if the trial continues to look safe and efficacious, it's easy to predict that perhaps the trial's cohort will be expanded to include patients who recently had a heart attack or patients with a family history of heart disease. And if that continues to look promising, then perhaps well, there are 8 billion people on the planet that have PCSK9. Genetics already tells us that there are people with natural mutations in PCSK9 who can eat all the Cheetos they want and they never get high LDL cholesterol because of their mutation. And this really is a way of using base editing to recreate that situation in any patients who want that benefit and would stand to benefit from having lower LDL cholesterol, which I think turns out to be a lot of people. So that's just one example. So in other words, I think disease prevention will eventually become one of uh, the largest impact applications of therapeutic gene editing. First, of course, I'm an academic, so I dream a lot. First, we have to address the patients who are at the biggest risk of death and poor quality of life because of their genetic disease, because in these patients, we have the highest risk to benefit ratio, or the highest benefit to risk ratio, I should say. Um, but I think, one of the great services that these patients like baby KJ, like Tracy Atterbury, like Alyssa Tapley have done is not just pave the way for other patients with their disease, but pave the way for regulators, scientists, doctors, and society to become more comfortable with the idea of carefully exerting our ability to control our genomes as a way of improving the quality and the length of our lives.
Now, I wanted to ask you about the impact of the federal funding cuts and the funding freeze to Harvard, changes in how grants are administered by NIH on your lab. What if any, impact has your lab seen from that?
Yeah, so first I'll say that many of my colleagues have been impacted much more than our lab. So I feel almost bad articulating the ways in which our lab has been impacted, but it has been impacted. All of the training grants that support many of the graduate students in my lab were terminated. The student fellowships, these NSF fellowships that some of our hardest working and most talented, most gifted graduate students work very hard to try to earn. They're honorific. They support part of their stipend. They were suddenly terminated. These are not fellowships that actually cost the government that much. They mean a lot to the graduate students. They were all terminated. So it was it was the better part of $1 million of funding a year that to the trainees of our lab that suddenly disappeared. But I think more importantly, there are many colleagues in the Harvard ecosystem who suddenly, through no real fault of their own, found their research, which has nothing to do with a political issue, terminated. And I know colleagues who have lost a third or half or 2/3 of all of their lab's funding. And as you might imagine, that creates a situation where you suddenly have to decide which projects, which people, which mouse colonies, which monkey colonies in some cases, you are going to continue to advance versus you have to let go of.
Mhm.
And that's heartbreaking because you can't start research projects and stop them and start them like you can start and stop a car. It takes years to set up projects so that they can be rigorously done to have hopefully a lasting impact on science and medicine. And then finally, I guess I'd say that I've never seen in my 26 years as a professor here our young people question more, be uncertain more than they are now about the future of their own jobs, the future of pursuing a career in science or in medicine. And that worries me deeply because the lifeblood of the whole scientific enterprise are these incredibly young, talented students, including students from overseas that until recently, the United States has been able to recruit the very best and the brightest. And they want nothing more than to come to the US, contribute their gifts and their talents to advance science here in the United States. And the vast majority of them will stay. And now, of course, they feel this pressure of being unwelcome as scientists. This includes graduate students on F1 visas drawn from the very best students in the world. To get into a graduate program here as an international student, you have to be one of the best 1 or 2 or 3 students in your giant university overseas. And I feel so fortunate that places like Harvard are able to recruit these students who have over and over for decades proven to be among the hardest working, most effective researchers. And I think it's just shooting ourselves in the foot to not make them feel as welcome as possible and to foster their participation in US science, in US industry, in training other scientists, and in advancing these breakthroughs in science and medicine to the greatest possible extent.
What do you think will be the long-term impact of these changes in funding on the kinds of advances that you're working on science in general? I mean, even on New England, which is such a biomedical powerhouse.
Well, it won't be good for sure. It slows down research. In some cases, it forces projects to shut down. In other cases, it incents professors to shut down their lab early. It makes it harder for companies to um, uh, find and recruit and maintain the talent and to do the fundraising they need to keep advancing science and medicine towards some kind of tangible societal benefit. And again, I think the biggest worry is that it discourages future generations of young scientists that we will rely on to serve as the lifeblood of this enterprise from continuing to devote the best, most creative years of their life to advancing science and medicine. You know, none of these people decided that the best way to make a big salary or to work the easiest possible hours was to become an academic scientist. Nobody enters graduate school to make money. So, you know, I think we really need to treasure and honor and encourage these people who have so much to give the system and are so willing to give the system their talent just because they want to have an impact on science and medicine. So that's really the aspect of the enterprise I worry about the most right now. And you know, hopefully, uh, cooler minds will prevail, and dialogue with the government and universities will advance in a productive way that allows us to protect these most precious uh, components of the scientific enterprise.
Okay. I guess one last question: is there anything that that gives you hope? Like, what gives you hope for
Is this
the
we better
resilience of?
end on a positive note?
Yeah, that's what I was thinking.
Well, I mean, to me, one of the defining traits of our species is this relentless desire to use our hard work and our talents and our gifts to make the future of our children and our children's children brighter than our own futures. That's just, to me, sort of one of the defining attributes of our species. Uh, it has never been a better time, a more capable time to use science and medicine in these incredible, bold, breakthrough ways, as your audience will hear about throughout the day, to save lives, to improve lives, to make us and our families better off than we were before these discoveries. So, science and medicine has never been more capable. I don't want the message of my concern about the future of science here to overshadow that key point. Science and medicine have never been more capable of improving our lives, have never demonstrated a greater impact to improve our lives. Uh, and that's just something we have to treasure and celebrate because it will play such an important role in defining the quality of all of our lives for the foreseeable future.
Thank you so much, Dr. Liu.
Thank you. A pleasure.
Thank you, everyone, for joining us.